Aspartate transcarbamylase, or simply ATCase, is an allosteric enzyme, and like most allosteric enzymes, it displays cooperativity. So in this lecture, I'd like to focus on the cooperative nature of ATCase and then discuss two allosteric effectors to this enzyme. So we're going to focus on cytidine triphosphate CTP and adenosine triphosphate ATP. Now let's begin by discussing the quaternary structure of this enzyme and how its quaternary structure actually affects the likelihood that the substrate will bind onto the active sites. Now, when the concentration of the substrate molecule is low inside the cell, the entire quaternary structure of the enzyme exists predominantly in the T state, the 10th state. So what do we mean by the 10th state? Well, in the 10th state of this enzyme, the two catalytic trimers, and these are the orange structures in this diagram, are basically found in close proximity with respect to one another. And they create a very compact and very constrained structure. And so what that means is the active sites in these catalytic trimers are going to have a low affinity for that substrate molecules. And so at low substrate concentrations, the substrate molecules are not going to be likely to be bound to those active sites, and so that will create a low catalytic rate, a low catalytic activity. Now, when we begin to increase the substrate concentration inside the cells of our body, the substrate molecules will begin to bind onto the active site. So let's suppose one substrate molecule binds onto one of the active sites of this enzyme. And once the binding takes place, what that does is it shifts the equilibrium of this entire equation slightly to the right side, from the T state to that R state. And so as we increase the concentration of the substrate even more of those substrate molecules begin to bind onto the active sites, and eventually that shifts the entire curve to the right side. And at a very high substrate concentration inside our cells, the entire curve will be shifted this way. This area will be very, very small, and so this enzyme will exist predominantly in the R state. Now, what do we mean by R? What do we mean by the R state? Well, the R state is also known as the relaxed state, and that's because these two orange catalytic trimers have now moved far apart, they rotated, and so they created this relaxed structure. And in this structure, the active sites have a high affinity for the substrate molecule, and so what that means is the rate of activity of this enzyme when the enzyme's quaternary structure is in the R state will be much higher than compared to that T state. And that's exactly why at high concentration of substrate, the activity of that enzyme will be high because it exists in that R state. So what's actually taking place is, upon the binding of the substrate molecules into the active sites found on the different catalytic trimers basically begin to interact with one another, and they cooperate with one another. And that's exactly what shifts the entire curve from one side of the equation to the other side of the equation. And this is what we know as the cooperative behavior of allosteric enzymes, such as aspartate transcarbamylase. And this mechanism can be described by using the concerted model, which we actually use to describe the behavior of hemoglobin molecules. So based on this model, the activity of this aspartate transcarbamylase is all or nothing. It either exists in the T state, in which this enzyme is inactive, or it exists in the R state, in which the enzyme is fully active. And it's the concentration of the substrate inside the cell that ultimately determines where the equilibrium will actually lie. At a high substrate concentration, the enzyme's quaternary structure will exist in the R state, while at low substrate concentrations, the enzyme's quaternary structure will exist in the T state. Now, if we take a look at the following diagram, we have three different curves. So the y-axis is the rate of product formation for this enzyme catalyzed reaction, and the x-axis is the concentration of that substrate molecule. Now, at low concentration of the substrate molecule, we're going to exist predominantly in the T state. And it's this green curve that describes the Michaelis-Menten curve for this particular enzyme, for this particular quaternary structure. And so notice, at some particular substrate concentration, the rate of activity will be low, and that's because the active sites of the enzyme have a low affinity for that enzyme. But as we begin to increase the concentration of that substrate, that begins to shift the entire curve to the into the R state, and once we transform the quaternary structure from the tense to the relaxed state, the blue curve is the curve that basically begins to describe that quaternary behavior. And so according to this blue curve, at the same substrate concentration, we see that the rate of activity is much higher. So the green curve describes the Michaelis-Menten curve for the tense state, the blue curve describes for the relaxed state, and if we combine these two curves, we obtain the actual black curve that describes the S-shaped, the sigmoidal curve for this allosteric enzyme. And this is what we know as the concerted model. We have the all or nothing behavior in which we either exist in the T state, in which the enzyme is inactivated, or the R state, in which the enzyme is fully active and basically carries out that particular catalytic reaction. Now, what about the two types of allosteric effectors that our cells actually use to basically control and regulate the activity of this enzyme? So let's begin by focusing on CTP. So remember, this enzyme catalyzes the first step in the biosynthetic process of CTP, cytidine triphosphate. And it's the CTP that basically creates a negative feedback loop that goes back and binds onto the allosteric sites found on these red regulatory chains. And by binding onto these red regulatory chains, it decreases and inhibits the activity of that enzyme. The question is, how does this actually take place? Well, upon the binding of CTP to the red regulatory chains, what it does is it stabilizes the T state of that enzyme. So it lowers the energy of that T state, it makes it more stable. So as these brown CTP molecules bind onto each one of these regulatory chains, as shown here, it stabilizes the energy of this T state, makes it much more stable. And that shifts the equilibrium from the R state into the T state. And so what that means is when CTP actually binds onto the regulatory chains, these CTP molecules make it more difficult for the substrate to actually bind onto the active sites and transform this quaternary structure from the T state into the R state. And so in this manner, our cells use these CTP molecules to actually regulate the behavior of this enzyme. So when we have a high concentration of CTP, we don't want to produce any more CTP, and so the cell uses the CTP in a negative feedback loop to basically inhibit the activity of this enzyme. Now, the second type of allosteric effector of this enzyme is actually ATP, adenosine triphosphate. In fact, adenosine triphosphate, just like CTP, also binds onto that same regulatory chain, these red chains. But unlike CTP, when ATP binds onto these regulatory chains, it actually increases the rate of activity of this enzyme. And it increases the rate of activity of the enzyme by displacing the CTP molecule from that regulatory chain and shifting the entire curve from the T state to that R state. So adenosine triphosphate, or ATP, is also an allosteric effector of ATCase. It binds to the same site on the regulatory chain, but it actually increases the activity of that ATCase. So at high concentration of ATP, the ATP will displace the CTP from the regulatory site on those regulatory chains, and that will shift the equilibrium from the T state to the R state and make the active sites much more likely to actually bind and convert that substrate molecule into the final product, the CTP. Now, the question is, what is the physiological significance of this effect? Why exactly does a high concentration of ATP activate this enzyme in the first place? Well, for two reasons. First of all, if we have a high concentration of ATP, that means we have plenty of energy inside our cells. And that implies if we have plenty of energy, we can easily use that energy to actually synthesize these nucleoside triphosphates, such as ATP, by using this enzyme right here. Now, the second reason why a high ATP concentration basically stimulates this enzyme to carry out its process is because of the following reasoning. So at a high concentration of ATP, when we have many ATP molecules inside our cells, that means we can use the ATP molecules to actually synthesize nucleoside triphosphates, so RNA and DNA molecules. But to actually synthesize these nucleic acids, we not only need the ATP, we also need other nucleoside triphosphates, specifically pyrimidine nucleoside triphosphates, such as CTP. And if we have a high amount of ATP, that usually means we have an unequal distribution of the pyrimidine to purine nucleoside triphosphates. And to equalize this distribution of nucleoside triphosphates, pyrimidine to purine nucleoside triphosphates, we have to carry out this reaction to actually increase the amount of CTP, the pyrimidine nucleoside triphosphate, that is found inside our cells. So physiologically, a high ATP concentration means that there is an unequal distribution of the nucleoside triphosphates, the purine to pyrimidine nucleoside triphosphates, and therefore the cell tends to equalize this distribution by producing more pyrimidine nucleoside triphosphates, such as CTP. So we basically conclude that the reason this enzyme displays cooperativity is because the individual subunits of this enzyme actually interact with one another. And by binding of the substrate molecule, we essentially shift the equilibrium from the T state to the R state. Now, in order to regulate the activity of this enzyme inside our cells, in order to actually turn on or turn off the activity of the cell of this enzyme, our cells use these two regulatory effectors. CTP and ATP bind to the same regulatory chains, those allosteric sites found on these red regulatory chains, but they have opposite effects. CTP creates a negative feedback loop that basically decreases the activity, inhibits the activity of this enzyme, but ATP binds and displaces the CTP out of that regulatory chain, and it actually increases the activity of the enzyme by shifting the equilibrium from the 10 state to that more active R state.